nitrogen isotopes to partition N2O emissions in agriculture. My name is Kate Dennis, and I'm a Senior Marketing Manager here at Picarro for our science products. And today I'm excited to be joined by two speakers, Professor Richard Farrell and my colleague Nabil Saad. So Professor Farrell is joining us from the University of Saskatchewan up in Canada. He is an associate professor and the strategic research chair for the Ministry of Agriculture up at the university. He leads an interdisciplinary team of scientists that are looking at applied and fundamental research that's focused on the biological aspects of the soil-water plant continuum, in particular as it relates to nutrient supply and efficiency. Professor Farrell owns and operates 4 Picarro instruments for measuring isotopes in N2O, CO2, and methane. Today, he's going to be talking to us about his research on using nitrogen isotopes to partition direct and indirect emissions of N2O from N15 labeled crop residues. Following on from Professor Farrell's part of the webinar today will be my colleague Nabil Saad. Nabil is a Senior Product and Applications Manager here at Picarro. He has a background in isotope geochemistry with a PhD from Cornell. In particular, Nabil, while he's been at Picarro, has done a lot of development of our front-end extraction devices for our carbon isotope systems. More recently, he has become the manager of our mid-IR platform, so that's the platform that will be that Professor Farrell has used in his research to measure N2O isotopes. Today, Nabil is going to discuss the Picarro G5101I for high-precision measurements of delta N15 in N2O. Before I pass it over to our speakers today, just a couple of housekeeping items. We are recording this session, and we will make the recording available to all of you afterwards so that you can review the slides and the presentations. In addition, at the end, after both Professor Farrell and Nabil have had a chance to speak, we will have a Q&A session. It will probably be about 10 to 15 minutes at the end. If you would like to submit a question to that session, please go ahead and use the question tool on your GoToWebinar control panel. And with that, I'm going to pass the webinar over to Professor Farrell for him to lead his part of the discussion.
Okay, thank you, Kate. Okay, so these first couple of slides just sort of present context for what we're doing, my research group, and sort of why we're doing it. And if you're interested in N2O, you know that it's a significant agriculture is a significant contributor to atmospheric N2O. And then normally, at least in Canada, about 25% of all the agriculture-based N2O emissions are a result of the use of N fertilizers, but that about 1/5 of all the emissions are attributed to the N in crop residues, so during the decomposition of these residues. And the one thing is that IPCC applies the same emission factor for both fertilizer and residue N, so they consider them both to be sort of the same availability. And it doesn't matter whether you're using the IPCC default value or whether you're using a region-specific value, such as the ones that we use here in the Saskatchewan, which is at the northern end of the semi-arid northern Great Plains. But there's a lot of data out there that shows that there's, because crop residue is cycled slower, that this sort of slow cycling can protect against some of these N losses, and that in turn might lessen N2O emissions. But of course, this all depends upon the quality of the residue, so the carbon-nitrogen ratio, lignin content, a variety of soil and environmental factors, and as well as management factors for how the residues are put back into the soil. So this is just a figure that sort of, it's from an article written by Chen et al. In 2013 that sort of shows the possible impacts of crop residue amendments on soil N2O emissions. And on the left-hand side of the screen, it's sort of under aerobic conditions, and the right-hand side is under anaerobic conditions. And basically what happens is, first of all, the residue itself is just an organic N source. So by adding residue to the soil, you put organic N into the soil, which then decomposes, mineralizes, provides available N, which goes to N2O. And what they found in their meta-analysis was that the adding residues added about 12%, increased N2O emissions by about 12% compared to not putting the residues back into the soil. At the same time, it depends upon the quality of the residues, like I said, and for residues that have a high carbon to nitrogen ratio, that can sort of promote the microbial assimilation of the N that's in the soil and that's being released in the residue as it decomposes, which then removes some N from the available pool and actually decreases the N2O emissions. And at the same time, if we go into anaerobic conditions, it adds, the residue not only adds some nitrogen, it also adds carbon and energy to the system, and you can get increased microbial activity, which then in turn results in, again, higher N2O emissions. But if we push the system too far, we go, increase the anaerobicity of the soil and remove oxygen from it, we can push the system so far that all the N2O that's produced actually goes all the way, is reduced down to nitrogen, and so it's removed from the system, which then again lessens the N2O emissions. So there's quite a complex mixture of things happening in the system at any one time. So the challenge for us is how we separate out the contributions of residue N from the soil N and fertilizer N. And what we've chosen to do is use an N15 tracer technique where the substrate, in this case the residue, is labeled with 15N, and then the movement of the isotube through the system is monitored using the Picarro G5101I isotope analyzer. And the objectives of the study itself were basically to quantify and compare total N2O emissions and 15 N2O emissions associated with the decomposition of different crop residues, and to partition them out and to look at the direct residue-derived emissions. So this is the N that's in the residue that makes its way to N2O, and the indirect emissions, which would be the effect of the added carbon on the nitrogen that's already there. And basically it produces an indirect soil emission. And ultimately, we're just trying to get a better understanding of the role that residue decomposition can play in nitrous oxide production and emission. So this is just, the standard leaky pipe diagram of the pathway, microbial pathways for the production of N2O. And I just put it on here to sort of basically highlight the pathways that we're looking at. We didn't follow everything in the pathway. We were looking at taking the residue and through nitrification or denitrification out to N2O. So we weren't really looking for the complete reduction or NO or the individual pools of nitrate and ammonium, basically just following the N2O. And this is a soil zone map of Saskatchewan. And just a little context here, in the center of this map right here is Saskatoon, which is where the University of Saskatchewan is located. So we're in the dark brown soil zone. And it goes from the dark gray, black, dark brown, and brown soil zones. And so this is sort of cool and moist, and we get hot and dry as we move down to the southwest. So what we did is we collected soils from 4 of the zones where the major agricultural zones, the black, gray, dark brown, and brown soils are. They were all collected from long-term research farms. They'd been in continuous wheat for at least 10 years, so that they all had the same residue history and background. The soils varied in texture from loam to a silty clay. They varied in organic carbon from about 6% to about 2%. And most importantly, they varied in the amount of nitrogen, available nitrogen. So the black soil has about 62 milligrams per gram of available nitrogen, equally split between nitrate and ammonium. And the gray soil has about, it's closer to about 69 or 70 milligrams, and it's mostly nitrate. And then the brown and the dark brown have considerably less available nitrogen in them than the other soils. These were all done as an incubation studies, so it was done in a growth chamber. And what we did is we did it at 2 soil water contents. One was at 50% water-filled pore space, which was designed to sort of promote nitrification, and one at 70% water-filled pore space, designed to sort of promote denitrification. And our end sources were an unamended control to give us the background emissions. We had a fertilizer control that had a little bit of 15N labeled urea in it just as a reference treatment, and then 4 crop residues, pea, wheat, canola, and flax. And then each treatment combination was replicated 4 times with all of the microcosms arranged in a completely randomized design. Now, the first stage in all of this was to get the crops labeled. And so what we did is we grew crops in a small greenhouse that had a C13 depleted atmosphere. This is, we pumped in CO2 using a CO2 generator off of a propane tank so that the atmosphere in the greenhouse had a delta C13 value of about -20 to -25 per mil, as opposed to the normal atmosphere, which would be about +8 or +9. And that resulted in residues that had delta C13 values in the range of about -40 to -45. At the same time, we added labeled ammonium nitrate to the soil. We put in about a 10% enrichment, and then added fertilizer during the growing season for these crops over about an 8-week period. And that resulted in N15 enrichments that ranged from a little over 1.5 to just about 2.5% atom percent. And the other important thing here is that the residues were added to the soil based on how much we would normally see in from a field study or in a cropped field. And so the shoot-to-root ratio varied for them. So there's for flax, it's mostly above ground material, and for the canola, there's a lot higher below ground biomass. This slide is here just to sort of give you an idea of how much N was added. So we added the residues to all of the soils, and each soil got the same amount. So for urea, there was 10.65 milligrams of available N in all of the soils, and the canola was 63.36 milligrams of N in all of the soils. The same thing for the total here. These vary from soil to soil because of the amount of N that was present in the soil to begin with. And the available N had a much bigger impact on the total N than it did on the 15N. Now, the systems were set up. We used these media bottles as the sort of incubation vessels. The residues were ground, coarsely ground. They were mixed with the soil, brought up to about 30% water-filled pore space. Then they were packed into the jars to a bulk density of about 1.15. Then the jars were sealed. They were covered in aluminum foil to sort of keep the light out, keep them dark, and incubated at room temperature. Then we would collect headspace samples. So we collected them frequently at first, so it was about every 12 hours for the first 3 days or so, and then the intervals increased as the experiment progressed. And what we would do is we would take out headspace samples, inject it into a serum bottle, because we needed a large volume for the Picarro instrument, the way we were operating it. Then we would take a small sample from this serum vial, put it in an excitainer vial, and analyze it for total N2O using a GC with an ECD. The isotope analysis was done. The samples were analyzed using the Picarro G5101I isotopic N2O analyzer to obtain the isotopic signature of the N2O. From this, we would calculate the 15 N2O concentrations, calculate daily and cumulative emissions,